Hello and welcome back to Microbiology. This is going to be Chapter 7 over nutrition, ecology, and growth of microbes. Microbial nutrition is going to be an important thing we need to discuss, but before we go too far, let's point out what nutrition is. Nutrients are going to be things that are basically acquired from the environment. Essential nutrients are things that we can't typically make ourselves and must take from the environment. Macro and micro are pretty self-explanatory. Macronutrients are things we need large amounts of, while micronutrients are things we only need small amounts of. Whenever we look at this, we can also look at it as inorganic versus organic. Inorganic means we're not going to typically have both carbon and hydrogen, while organic is going to have both carbon and hydrogen, and usually are going to be products of living things. So you can see here we have a primary list of resources we need for life and where we would find them in the environment. Naturally, again, living things would have these inside of them, so consuming other living things is a good way to get them. Here we see a breakdown of a E. coli cell, and what we're looking at here is really three different tables. The first table is going to show us the concentration of things like proteins, RNA, DNA, carbohydrates, lipids, and the miscellaneous stuff. Notice we have both total weight and then dry weight. Total weight illustrating with water. So with water, proteins are 15% of the cell, RNA 6, DNA 1, carbohydrates 3, lipids 2, and miscellaneous 2. But notice whenever we eliminate water, proteins become half the cell. RNA becomes 20%, DNA becomes 3, carbohydrates are up around 10, and so are lipids because they're energy storage. So the idea here is water is quite important to the cell. But as we look, water is also going to be just a portion because really proteins, RNA, DNA, carbohydrates, lipids are quite necessary as well. So if you look again, the bottom image here or table is showing you the percent total weight. So water actually makes up 70% of the cell. Over here we're looking at dry weight in terms of percent. So this is again not counting water, but notice how high oxygen and hydrogen are. Okay, that's still quite abundant, even not considering things like water. The next thing we're going to start discussing is where do we get our carbon. And for heterotrophs, carbon comes from really consuming other things, because again, life will produce organic carbon, and that's what heterotrophs need, is organic carbon. Autotrophs, on the other hand, will use inorganic carbon and are often called self-feeders because they can convert that CO2 into ultimately things like organic carbon compounds. Nitrogen is another very important resource that the cell needs. Nitrogen is used in things like DNA, RNA, proteins, and even ATP. So we need a pretty steady supply of nitrogen. Unfortunately, a large amount of nitrogen out there is not something that's readily used by living things. And as a result, we often need things like nitrogen fixers or denitrification organisms to take and allow us to use a lot of the nitrogen in the environment. Oxygen is going to be necessary to do lots of things, including things like carbohydrate production, lipids, nucleic acids, and even proteins. Not to mention it's also part of water, so oxygen is routinely needed by the cells. Hydrogen is going to be another very important resource. Again, hydrogen used in water, which we've already stressed is very, very abundant in the cell, but it's also used to do things like hydrogen bonds. It's also a big player in pH, as well as things like the oxidation reduction reactions necessary for respiration. Phosphorus is another resource that we use quite a lot of because phosphorus is going to be used for things like ATP and even in some situations things like amino acids and genetic material. Sulfur is one that's going to be a little less important. It's not used in a lot of things, but it is used in something called methionine, which is one of the major players in terms of amino acids. In fact, methionine should be the first amino acid added to any chain because guess what the start codon codes for? It's methionine. So there are resources that we must have in order for the cell to live, and so we call those resources growth factors. So it may be things like amino acids, nitrogen bases, or even vitamins. So again, these resources should be picked up from somewhere else in the environment that allows the organism to grow. The next topic we're going to cover is phototrophs and chemotrophs. The question here is, where are you going to get your energy? With phototrophs, you're going to get energy from the sun and do photosynthesis. With chemotrophs, you're going to gain energy from eating other things, again, breaking them down so that the chemicals are what fuel your actual energy needs. So we can actually combine all those troph words, including photo, auto, chemo, and hetero. So let's start off with the photoautotroph. The photoautotroph is going to get energy from the sun, hence the word photo. But autotroph meant that it got carbon from the air as inorganic carbon. So this is really, again, an example of plants. Chemoautotrophs mean, again, energy from chemicals, but autotroph was inorganic compounds of carbon, and so what we see here are really deep sea bacteria or archaea that can do this, where they're feeding off of the chemicals but also using the inorganic carbon spewing from the deep sea vents. The next category is the photoheterotroph. So again, sunlight for energy, and then organic carbon. And really, these are purple and green photosynthetic bacteria. And then the last category that we're going to look at here is just the chemoheterotroph. Chemoheterotrophs are going to use chemicals for energy, but again, organic carbon. This really applies to us and many other things around us. There are the two subcategories, parasite and saprobe. Again, parasites feed off the living and saprobes feed off the dead. So we just kind of went through this. Again, photoautotrophs, very important for the food web. They're the primary producers that help support all other life. Chemoautotrophs, typically pretty important whenever it comes to being producers in environments where sunlight isn't readily available. And that's why, again, the deep sea environment, they're a primary producer for it. Methanogens are going to be basically methane-forming microbes. They do exist often in pretty extreme environments where oxygen is basically non-existent. And so the idea is we could produce a methane waste product. Methane is relevant because it is a significant greenhouse gas, but also very flammable. Chemoheterotrophs are what many of the things around us are. Again, chemoheterotrophs use chemicals for energy and organic carbon. So, like us, they typically eat or break down other things for their energy and carbon needs. So, aerobic respiration we did in Chapter 8. Again, we're not going to go over it extensively again, but do understand that aerobic respiration is the complement to photosynthesis, that the waste from aerobic respiration is what really fuels photosynthesis, and the waste from photosynthesis is what we use to do aerobic respiration. Saprobes, again, are decomposers. Most of the saprobes out there really are bacteria and fungi, and their job is to really break down resources in the environment so that we can recycle nutrients and elements to allow other life to continue. Parasitic organisms are going to exist as well, but they're different from saprobes in that they feed off the living. In this case, again, parasites will actually steal resources from the host, hurting the host. These are all considered to be pathogens, and they really fall into four categories: the ectoparasites that live on the body, the endoparasites that live within the body and tissues and organs, the intracellular parasites that live inside of cells, and then the last category is obligate parasites that must be in or on a host to live. Again, without having a host, they cannot live. So let's talk about how organisms actually feed, because we're going to have to discuss nutrient absorption, which is how we would pull resources from the environment and transport them into the cell. Transport is going to happen across the cell membrane and typically requires specialized structures to do this. We'll discuss several of the options as we go forward. The first technique we'll discuss is diffusion. Diffusion is going to be a natural process that does not require additional energy. But what happens with diffusion is we'll always go from a high concentration to a low concentration. So use this example with the sugar cube in the tea or coffee or whatever it is here. Again, the sugar molecules will want to spread apart to areas where there's less sugar molecules. That would be an illustration of diffusion. We can speed up rates of diffusion by adding energy into the system. So again, if this is hot, the molecules will move more quickly. Again, if we stir, we're adding again a degree of increase of energy. And so the idea is diffusion will happen naturally if enough time is given, but diffusion rates can be pushed based off of concentration. So one type of diffusion that we should talk about is osmosis. Osmosis is going to be the movement of water across a selectively permeable membrane. Typically, if a resource like a solute can't move across the membrane, it's much easier for us to move water across instead. This illustration is showing us how osmosis would work. Notice inside the semi-permeable sac we have actually large amounts of red balls or solute. Since the solute can't move through the red bag, the only thing that can happen is we can move water into the bag to dilute the solute. In doing so, notice that water becomes more abundant and the level of water actually increases up the tube. This is really defying gravity because the need to dilute that concentration of solute is so high that the water will continually rush in to do so. So this leads us to our isotonic, hypotonic, and hypertonic conditions. So let's start off with isotonic, where the concentration of solute inside and outside are roughly equal. And as a result, water really isn't going to move very much in terms of major amounts. What we'll see is roughly the rates of water moving in and out are about the same, which means there's really not much net change. In the hypotonic condition, we're going to have a situation where the solute concentration inside the cell is going to be much higher than outside the cell, and as a result, water would want to rush into the cell to dilute the solute, ultimately causing the cells to potentially swell and burst. Hypertonic is the exact opposite of this, where there's much more concentration of solute on the outside, and as a result, water will leave the cell to dilute the solute on the outside. This again typically would cause the cell to want to shrivel up or collapse in upon itself. So we see this illustration of all three of these scenarios. Notice in isotonic that there's roughly equal amounts of solute inside and outside, and as a result, water levels stay consistent. This is the ideal situation for most cells. We want to be in this situation because now, again, we don't have any concerns of death. In a hypotonic, notice the concentration of solute inside is much higher than outside, and as a result, water wants to rush in and potentially cause the cell to swell and burst. In the hypertonic situation, notice the concentration of solute is much higher outside. Naturally, again, water wants to go dilute the solute, causing the cell to typically shrivel up. As I stated before, an isotonic situation is the ideal situation for most cells. They want to live in an environment where, again, water isn't going to be a concern rushing in or rushing out of the cell. In hypotonic situations, there can create issues. One of the things I want to point out that can help protect a cell against swelling and bursting is actually that cell wall. So cell walls are really, again, going to help often with changes in tonicity. The hypertonic, again, the concern is the loss of water. There are some bacteria that have the ability to actually take salt into their cells that allow them to now create more of an isotonic environment. So let's talk about now facilitated diffusion. This is still diffusion and still natural, just like other forms of diffusion and osmosis. But with this time, we're going to have to invest a tiny amount of energy in making carrier proteins. But once the carrier proteins are in place, no energy will be required to move things in and out of the cell. So with these carriers, they're going to allow for now movements, but movement with specificity, making sure we're moving the things that we want in and out across the cell membrane. So this is an illustration of the different types of passive transport processes in cells. And whenever you see passive transport, think that there's no need for additional energy like ATP. We don't need ATP for simple diffusion to occur. Again, whenever we see this, we will always go from a high concentration to a low concentration. Same idea happens down here with facilitated diffusion, but this time we have a carrier that now helps us control what is moving across the membrane, but we still want to see a balancing of concentration on either side of the cell. There is something called saturation because the transport rate of a substance is limited to the number of sites through which we can transport. So if we only have a certain number of those transport proteins, we can't move any more substrate than what the number of transport proteins can. And so there can be a situation where literally, even though we have huge concentrations, we're stuck moving the basically solute at a slower rate because we don't have any more pathways. Competition is going to be another important idea. Just like we did before with competition, if there are two molecules that want to buy or try to fight for the same spot, only one can really go at a time, and as a result the thing with the higher binding affinity would move much more quickly than the thing that had a lower binding affinity. So let's talk about active transport now. Whenever you see the word active transport, you should immediately think of ATP, because we're going to need ATP to make this work. The other thing I want you to understand is this is going to be going against nature. This is not that balance that nature wants. We're typically going to create imbalance for reasons that would often lead to things like metabolic activity. So these are things like pumps, where we would physically pump things out, such as ions like the sodium and potassium and even hydrogen ions across the membrane. Another type that we would look at here is group translocation. With group translocation, we're going to really move several things, but in the movement we're going to convert them to a substance that is ready to be used inside the cell. The best example of this is how we move glucose across and really do glycolysis in that step of group translocation. Endocytosis is another example of us moving molecules across with ATP, but since we're moving large molecules or large amounts of things, we're going to have to invest energy. There are two major types, phagocytosis and pinocytosis. With phagocytosis, we're going to eat things, so you bring in solids. With pinocytosis, pino means to really drink, and as a result, we'll be bringing in liquids, things like oils. This is an illustration of active transport processes. Again, these require ATP. There are really three of them located here. Things like carrier-mediated transport is really kind of like that idea of pumps, where we can push a resource to one side of the membrane, creating a concentration. That concentration can be used to do lots of things like metabolism. Group translocation is going to still be, again, movement across to typically create concentration, but notice how the things that are being moved are modified as they come through. The best example is, again, that process of glycolysis. As we move glucose into the cell, we're already taking care of glycolysis. And then we have our bulk transport that include phagocytosis of solid things and pinocytosis of liquids. These things will again be moved inside of vesicles and transport typically towards the center of the cell. So let's now talk about environmental factors that influence growth of microbes, including things like temperature, pH, gas availability, radiation, even things like pressures. So the first thing we're going to discuss is cardinal temperatures. Specifically, the cardinal temperatures illustrate the minimum temperature where we still see growth, the maximum temperature, the highest temperature before the cell dies, and then optimum temperature where the cell would grow best. With the minimum temperature, this is really the lowest temperature where the microbe is still reproducing. Under the minimum temperature, though, we're not typically seeing the cell die. It just becomes inert. It isn't doing anything. And so there are lots of situations where we actually can lower temperature to decrease growth. And that's really how refrigeration works. If we bring temperatures down, microbes don't want to reproduce and spoil your food. Maximum on the temperature is the highest point where the cell is still alive. Going beyond maximum temperature results in death as we destroy enzymes and proteins, causing the cell to typically die. Optimum temperature, though, as the name implies, suggests this is the place where the growth rate for the bacteria will be the highest, meaning they're most effective at this very specific temperature. Whenever we look at this, notice that we recognize minimums and maximums. minimums being the lowest temperature where we see still growth, and maximum being that point where death occurs. So you can see the maximum being that death point. Notice though the high point is really going to be that optimum temperature. So if we look at this green line, notice the minimum temperature falls around 10 degrees Celsius. The optimum is going to fall around 37 to 40 degrees Celsius, and then our maximum is going to be around 47 degrees. Notice how much closer the optimum is to the maximum. That should illustrate that many things live very close to their thermal death point, which is potentially bad for them. That should make sense why fever could even be enough to cause the death of some microorganisms. But the bigger idea here is notice that we have different colored lines. Different microbes like different environments. Their minimum may be different than our minimum. And so the idea here is that things that like it cold we call psychrophiles. So blue line illustrates a psychrophile. Psychotrophs are kind of in the middle. Really don't see a lot of them that we care about. The mesophiles, on the other hand, are something that we are concerned with because this is the optimum temperature for us as humans as well. Those thermophiles will like it hot, as the name implies, and then the extreme thermophiles like it really hot. Notice we're well above 100 degrees Celsius here, meaning we love it above boiling if we're an extreme thermophile. Psychotrophs are crazy for liking it so cold. Again, they typically grow around 0 degrees Celsius or, again, freezing. The idea is they can't grow in the higher temperatures, but they do, again, still grow even though it's quite cold. Mesophiles like more of that moderate environment, much like we do. So depending on the organism, there can be kind of a range, but most of them do like to grow typically around 37 degrees Celsius, especially the ones that are pathogens for us. Thermophiles are going to grow at higher temperatures. Many of them actually thrive up around boiling. Gases are also going to be an important environmental factor we need to account for. Naturally, many things need oxygen to do, again, things like aerobic respiration. But many people don't realize there are dangerous versions of oxygen out there. So if an organism is going to routinely be in the presence of oxygen, it needs ways to detoxify it. And so the idea here is that we need mechanisms like enzymes that can break down oxygen if, again, it's a dangerous version of oxygen. So let's discuss some of those dangerous versions of oxygen, like singlet oxygen. A single oxygen is actually very dangerous because it wants to bind to something, and it's wanting to really take on 2 electrons. And so these are very reactive and can damage or even destroy cells. Superoxide ions, hydrogen peroxide, and even hydroxyl radicals are going to be also dangerous versions of oxygen. But there are enzymes that some things have to basically counteract this. So catalase is a good example. The job of catalase is to actually break down hydrogen peroxide. We'll actually split hydrogen peroxide into really hydrogen gas and water. Since we just talked about how we can remove dangerous versions of oxygen, let's talk about how some organisms require oxygen or no oxygen at all. So aerobic organisms are going to require gaseous oxygen to carry out metabolism. These things, again, do aerobic respiration. We will see some things absolutely require this pathway, so we call them obligate aerobes. But we also see something called facultative anaerobes. So let's start off with the word anaerobe. An anaerobe is something that does not require atmospheric oxygen, and so they can grow in the absence of oxygen in the air. But the word facultative is interesting because facultative really means kind of either or. So we'll give you a scenario. A facultative anaerobe can grow in the presence of oxygen or without oxygen. So the idea facultative is illustrating an either or scenario. So again, facultative anaerobes can grow with atmospheric oxygen or without atmospheric oxygen. But the microaerophiles are unique in that they still need atmospheric oxygen, they just need low amounts of it, small amounts, hence the word microaerophile. We need small amounts of air. The word anaerobe, though, illustrates we don't need atmospheric oxygen, and instead use different versions of oxygen and the anaerobic pathway. There are things called strict or obligate anaerobes. This means that they cannot be in the presence of atmospheric oxygen. If they are in atmospheric oxygen, they will die. One of the last ones we need to talk about here is aerotolerant. Aerotolerant anaerobes do not use oxygen, but they tolerate them. So specifically, they do not use the oxygen in the air, but they can survive in the presence of the oxygen in the air. So let's use these 4 tubes to illustrate what we just discussed. Notice in this first tube, we just have growth really at the top of the tube. This would illustrate an aerobe or an obligate aerobe. Notice in the second tube, we have oxygen really, or growth really right here where oxygen is available, but not in high concentrations. This is typically thought of as being something like a microaerophile. Notice in this tube, we see growth from top to bottom. There are really 2 possibilities for this. This could be a facultative anaerobe or an aerotolerant anaerobe. But here on the far right, notice we just really have growth at the bottom of the test tube. This illustrates again an anaerobe. There are some organisms out there that require higher than normal levels of CO2, and as such we often have to grow them in isolation and really pumping lots of CO2 into the incubator. pH is going to be another one of those environmental factors. Again, the word phile is going to illustrate to love something, so what precedes phile is what we love. So what do you think an obligate acidophile loves? The answer is acids. And notice since it has the word obligate, it must be in the presence of acid to grow. What about an alkalinophile? Alkalinophiles like very basic conditions. Osmophiles like situations where there's a high solute concentration in the habitat. Obligate halophiles, again, love high amounts of salt in the environment because they themselves are typically full of salt. That way they can live in a isotonic situation. But what about the facultative halophile? The facultative halophile is going to be able to survive because they typically can kind of change the amounts of salt concentration they have internally or externally. Radiation is another one of those environmental factors that can play a big role in whether or not we see growth. In general, most things routinely exposed to UV radiation have to find a mechanism to prevent it from damaging DNA. If they don't have these mechanisms, they will die if they're exposed to too much UV radiation. Barophiles are an interesting category because they rely on hydrostatic pressure to keep them put together. So they typically live deep in the water, where again there's significant amounts of pressure applied to their outer surface. What do you think would happen if we pulled a barophile out from the bottom of the ocean? Really, they'll fall apart. With barophiles, they need that pressure to keep them put together. So no pressure, and they fall apart. So far we've talked about how the environment and also nutrition can affect growth, but we haven't discussed much about the associations between organisms, which really fall into 2 categories, symbiotic and non-symbiotic. With symbiotics, they're going to live in close nutritional relationships and will typically either be required by at least one party, if not both. In non-symbiotic relationships, organisms are free living and their relationship is not required for their own survival. So let's start off with the symbiosis. A symbiotic relationship is trying to illustrate that there is a reliance on one or more parties for one's survival. So one of the first things we should point out is mutualism. With mutualism, both parties benefit from the interaction that they're having. A good example of mutualism would be this example of feeder fish. These feeder fish are going to rely on food resources from the bigger fish and ultimately then clean the bigger fish so that we can have a healthier existence without likelihood of disease or infection. Another good example of mutualism is the interactions you have with many of the microbes in you. Again, there are many parts of our digestive system that we can't produce all the resources we need, and as a result we rely on microbes to make those resources and finish out digestion for us. Commensalism is another situation where we're going to have one party benefiting from the interaction, but the other party is actually not at all affected. So in a commensal relationship, one gains a benefit, but the other is not hurt nor helped. So the best example I can think of for this one is the sea anemone in the clownfish. The clownfish gains benefit from hiding in the anemone. But does the anemone really gain anything from the clownfish being there? Really, no. And as a result, the clownfish gains benefit, but the anemone is neither harmed nor benefits from the interaction. The last symbiotic relationship I want to talk about is parasitism. With parasitism, one party benefits while the other party is physically harmed. As we've discussed before, hosts will provide resources to the parasite that keeps the parasite alive. But again, in providing those resources, it harms the host. So let's move on now to the non-symbiotic associations, including antagonism and synergism. With antagonism, there's going to basically be the situation where multiple members of the same community compete with one another. And so the idea is we can encourage or discourage growth based off of what organisms are present. So having good bacteria in our gut prevent bad bacteria from being there. That's an example of antagonism. Synergism is going to be an interesting relationship where, again, two or more free-living organisms actually gain benefit from one another being present. But again, they're not relying on one another for survival, and that's what makes synergism different from mutualism. Synergism just means we can get more done if we're together than if we're apart. Antibiosis is a form of antagonism, where in antibiosis we're going to produce a compound that specifically prevents the growth of other organisms. So antibiotics is a great example of this. Fungi produced antibiotics to prevent bacteria from growing, because if the bacteria were not there, they could not steal the food of the fungi. Biofilms are the topic we should cover next, because biofilms are really a great example of synergism, where multiple different organisms will come together to actually live and work more effectively as a unit. And so the idea here is lots of bacteria will come together to help one another survive. All these bacteria could live on their own, but since they're living together, they actually thrive more than they would on their own. So interestingly enough, biofilms actually form not in random. What happens is a pioneer colonizes initially, allowing for the production of typically lots of sugary materials that allows for other microbes to come in and grow amongst the pioneers. Really, this is an assembly done through something called quorum sensing. Quorum sensing is basically the release of chemicals that send messages to encourage other microbes to do specific tasks. So as you can see here, we have the pioneer establishing growth initially. They secrete resources and even, again, quorum sensing materials to the outside environment that encourages other bacteria to now join in. And before long, we have this giant mass of bacteria that are living and working together, and they're being very successful. But it's not uncommon to see some bacteria leaving to go start new biofilms in other places. The last thing we're going to do with our time in this chapter is discuss really what is bacterial growth. And we've defined this before, but bacterial growth is not an increase in size, but rather an increase in numbers. So we have to talk about bacterial replication. Bacteria reproduce through a process called binary fission. This is a very simplified version of what we see with PMAT. Since we just have one chromosome in bacteria, all we have to do is copy it and send one copy to each end of the cell, and then the cell divides in half, and now we have twice the amount of bacteria that we had before. So really, after each generation, the number of bacteria double. So we call this the generation time or doubling time. And what this can lead is to a situation of exponential growth, because after one generation, one bacteria turns into two bacteria. But those two bacteria after another generation turn into 4, and then the 4 turn into 8, and then 16, and then 32. Before long, numbers get very big very quick. And as a result, we have a significant problem with trying to basically graph the growth. If we were to try to use a normal graph, at some point the line becomes straight up and down or vertical. That doesn't tell us a lot. So in order to actually graph this, we have to do what we call a logarithmic graph. With the logarithmic graph, we actually get to see the pattern, because the pattern stays the same. Even though the numbers get really big really quick, All that happens is during each generation, we now see twice the amount of bacteria. And so we can see that pattern stays consistent. Even if numbers are getting very, very big very quickly, we still see that really this is just a doubling. So let's talk about the growth curve, because the growth curve is a nice illustration of how bacteria would grow in a normal population. So we can see in this growth curve that there are really going to be 4 steps. The lag phase is the initial startup, where 1 bacteria turns into 2, and 2 into 4, and 4 into 8. Again, numbers are still staying pretty low, but at some point numbers start to grow very quickly. We call this period of growth exponential growth, as again 1 million cells turns into 2 million cells, and 2 million into 4 million, and 4 million into 8. Again, numbers get very big very quick. That's why it's called exponential growth. But at some point resources become limited, in which case growth tends to plateau out. We call this the stationary phase. But whenever resources run out and waste starts to build up, notice we progress from stationary into a downhill slide known as the death phase. With death, we see again decreased numbers of organisms, because again resources are now becoming almost impossible to find. So again, the lag phase is that initial startup where numbers are growing, and we're still growing at the same rate we would, but since numbers are small, the numbers aren't getting big enough quick enough to see exponential growth. Again, what characterizes exponential growth is significant increase in numbers, but before long we can create an unfavorable environment, and that's whenever we see stationary growth. As resources run out and as again waste builds up, we start to see this progression into what's called death phase. And with death phase, again, the whole system starts to decline as microbes start to die because they can't find enough resources to live, and they're stuck in a bunch of waste that they don't like. Growth curves are important to us because they show us features like how antimicrobials would actually increase rates of death phase. If we put antibiotics in with a active or exponential growth, we may even skip stationary phase and go right into death phase. With the growth patterns we see in the basic development of a normal culture, again with that lag, exponential, stationary, and death, we start to see the same basic patterns whenever we look at infection. That during the initial stages of infection, we don't see many signs or symptoms, and it's because bacterial numbers are very low. But as bacterial numbers go into the exponential range, now we start to see those signs and symptoms. But hopefully, again, the immune system comes in and tries to stop this, and as a result, we start to see signs and symptoms plateau out and then start to decrease, so as we go from stationary into death phase. Since again it's important to note what stage of growth we're in, we need measurement methods to do so. So turbidity is actually a pretty effective way of determining the number of cells in the population. So we can use how cloudy a liquid media appears to determine roughly how many cells are present. That can again tell us what stage of growth we're in. But sometimes we need direct or really total numbers of cells present, and we call this a direct or total cell count, where microscopically we put the cells on a slide that has a grid pattern that we can then count the number of cells per grid and then multiply that to really get the exact number that we were looking for. So here's the whole turbidity idea. We can put it in a speck. The speck is going to again try to shine light through. If there are no cells present, then again the light will pass right through. But again, if there are a bunch of cells, it will scatter the light, so less light gets through and picked up, and ultimately that tells us again there are more cells. This is the example of the direct count. Notice again the gridded pattern. We can sit here and count how many cells are present in a squared area and multiply that by the number of squares we would expect to see to understand the total number that we would expect. Thank goodness for science and for technology, because we've made this new techniques that allow us to understand numbers quite accurately without having to do all those crazy tasks that we saw in the previous examples. So with the culture counter, this is going to electronically scan the culture as it passes through a tiny pipette. Each cell is going to be detected as a beam is broken, illustrating a number. So the flow cytometer, on the other hand, works in the same basic way, but it also has the ability to measure cell size and differentiate between both living and dead cells as well. With PCR, or polymerase chain reaction, we can actually try to quantify the number of bacteria based off of how many genes we actually find. And so the idea is we can use math and work backwards to estimate the number of bacteria that we started with. And the same logic is applied whenever we measure ATP output. We know roughly how much ATP a cell should make, and as a result, if we see so much amounts of ATP, we can work backwards to figure out how many cells we thought were there.